we can do that at some other time, perhaps. So genetics. Let's take that first one. And of course it matters what genes we inherit, and that's what we've thought about in terms of genetics for a long time as being important. But only 15% of the genes in our cells are turned on at any given time. So something must be going on that for those other 85%, they're just hanging out, waiting. Well, waiting for what? Well, waiting to be needed based on a reaction to the environment. And doesn't that make sense that humans have been so adaptable over the years that there would be a number of genetic cards, so to speak, that we have in our hands that we can play depending upon the environmental needs. Enough food, too much food, hot climate, cold climate, on and on. We've been able to roll with the punches because we've had choices of genes that we could actually turn on or turn off. The thing about this epigenetics, which is this turning on and off, these switches, if you will, for the genes, is that turning a gene on or off is not always a reversible thing if it happens at key developmental stages of life, such as we've been saying, prenatal and early life. And we actually know the body's on-off switches. And on this slide here is a kind of a cartoon description of that, and this is from a wonderful article in the New England Journal from July of last year. And if you want to turn a gene on, this is what we do. That blue curly line is DNA, and you can see that nothing in terms of gene sequence is being affected. That blue line is not being affected in any way in terms of no one's breaking it apart and inserting other nucleotides in there. It stays just the same. But what is changing is those orange football-like things up there are called histones. Histones are proteins that DNA winds around to kind of organize itself. If you take the DNA in any one cell and stretch it out, it's about 6 feet long. Just cram that down to a little tiny cell nucleus. Obviously, packaging is very important in terms of whether RNA can even get in there to transcribe that section of DNA. If you want to open up an area of DNA so that RNA can get in there, you put some acetyl groups on your histones. That's what that AC is up there. You also stick a transcription factor on that P box, which is your promoter area. These things will turn a gene on, so to speak. If you want to turn a gene off, you get rid of those acetyl groups on the histones, you get rid of the transcription factor, and you stick some methyl groups on the promoter area to kind of block that, because the promoter area is sort of like the ultimate on-off switch for the genes. There are methyl groups there. It completely blocks other things being able to get in there, and that gene is effectively turned off. So epigenetics then helps us understand, we've known for a while that this idea of nature versus nurture, which, the question has been asked which of those is more important. And we know actually that now that's an absurd question. It's not either or. It's that both of them interact with each other quite a bit. And as the study I'm about to talk about describes, nurture affects nature. This study was done in rats, and if you take rat pups that are raised by nurturing mothers, and if you're a rat, one of the ways you do that is you lick and groom your pups on a regular basis. And it's now been shown that literally the gene which affects stress hormone receptors gets turned on in rats that are raised by good nurturing mothers. And these rats grow up to be very stress-resilient rats. They're pretty mellow, the kind that if you put them in a new cage, they might just go, hey, this is kind of cool. I can look around and see what's going on here. Not stressed by it. But rat pups that are raised by neglectful mothers, the gene which affects their stress hormone receptors is turned off. These rats grow up to be very stress-reactive. You put them in a new cage, they will cower in the corner in fear. And you might say, well, how do we know that they didn't just get this inherited? They've taken rat pups that were born to mothers that were nurturing and mothers that were neglectful and reversed them so that they were actually raised by the other type of mother. Plus, they've actually been able to measure the DNA methylation on those genes to know which of these have been turned on or off. So as they say, there is no change in gene sequence. The changes are only in gene expression. Hence, these kinds of effects are called epigenetic, or above the genome, so to speak. Epigenetic mechanisms provide a potential pathway by which early experience can have lasting effects on behavior. And as Daniel Goleman says, it's biologically impossible for a gene to operate independently of its environment. Genes are designed to be regulated by signals from their surroundings.